Today I'm going to present the project Two Catalytic Fluoroquinolones. One of the main problems of the modern health is the resistance to antibiotics. Many types of bacteria show resistance to the known antibiotics, so we can't prevent or treat the infections they cause. More than 500,000 people die each year from drug-resistant infections, and according to the World Health Organization, this number will rise to 10 million in 2050. The problem is more serious because the development of the resistance is fast, and pharmaceutical companies don't want to spend money and time to develop new antibiotics that might be useless after a short time. So on the one hand, there are no new antibiotics, but on the other hand, the development of the resistance is rapidly growing. There are several approaches how to deal with the resistance problem. I'll talk about catalytic antibiotics. Catalytic antibiotic is composed of two components, binding domain and warhead domain. The binding domain, usually a known antibiotics, binds to a biomolecular target, a DNA or a protein. After binding, the warhead domain cleaves the target and totally destroys it. At the end, the drug is released and can do the cycle again and again. If we compare between non-catalytic drug to catalytic drug, we see that non-catalytic drug binds to one target and inhibits it, while catalytic drug binds to many targets and totally destroys them. What are the advantages of catalytic antibiotics and why we think they might solve the resistance problem? First, catalytic antibiotics cause rapid cell death and delay the development of the resistance. For example, the kill time of colifinis-3, a natural catalytic antibiotic, is 10 minutes, while the kill time of aminoglycoside, non-catalytic antibiotics, is 1 to 4 hours for the same target. Second, catalytic antibiotics should be more efficient against low binding affinity mutants. For non-catalytic antibiotics, low binding affinity deactivates the drug. This is a common mechanism of resistance. But for catalytic antibiotics, even when the binding affinity is low, if the cleavage is fast, the drug is still efficient, and thus overcomes the resistance. Third, catalytic antibiotics should be less toxic because they have high specificity to the target, both to the binding site and the cleavage site. Now I am going to present our work on catalytic fluoroquinolones. DNA gyrase and topoisomerase 4 are enzymes that control the interconversion between supercoiled and relaxed states of the DNA. The enzyme changes the topology of the DNA by double-nick the DNA so it can change its state, and then resealing the break. Fluoroquinolones is a class of antibiotics that binds to the enzyme and inhibits it by stabilizing the DNA enzyme complex, the ternary complex, thus causing bacterial death. We want to make catalytic fluoroquinolones. The binding domain is ciprofloxacin, one of the fluoroquinolones, and the warhead domain is copper and cobalt cyclane nuclease that can cleave the DNA in both hydrolytic and oxidative manners. We synthesize 6 compounds with different linkers between the domains. Our plan is that the cipro domain binds to the enzyme, and then the metal cyclane nuclease cleaves the DNA, so now the DNA is fully broken, and the enzyme is also ruined. The compound is released and can do the cycle again and again. We tested some biological assays. Most of them are based on gel electrophoresis to resolve the different forms of the DNA, the supercoiled, the linearized, and the nicked. We check DNA's activity to see if the compounds can cleave free DNA. Copper complexes show significant activity. We can see clear spots of nicked DNA. But cobalt complexes show no activity. There are no spots of nicked DNA. Then we did an enzymatic assay to figure out the mechanism. The stabilization of the ternary complex is demonstrated by the ability to generate linear DNA in the presence of proteinase K that hydrolyze the enzyme. We can see that if we cut the enzyme, we get linear DNA. We can see that for Cipro and the new compounds. We do not expect to get linear DNA without proteinase K. However, we get it for 4-copper and 2-copper. This is a proof of concept that 4-copper and 2-copper generate linear DNA in the presence of DNA gyrase, as we suggested, that the metal cyclase nuclease cleaves the DNA, so we get linear DNA without hydrolyze the enzyme. Unfortunately, we discovered that the DNase activity of the copper complexes is inhibited by amines as tris, as well as carboxylate and phosphate, due to irreversible water ligand exchange, demonstrating the vulnerability of the complexes. When we added DTT, a reducing agent, we saw significant improvement in MIC, the minimal inhibitory concentration, between the metal-free and metal complex. This is a proof of concept that 4-copper has oxidative activity ex vivo and in the presence of DTT. Our goal was to solve the vulnerability problem. We designed a dynamic cap that should protect the metal center and open close to the DNA to enable the catalysis. The chosen cap was amine. Its main purpose is to protect the metal center, as we said. It might also enhance the catalysis as a general acid and enable cobalt-mediated catalysis by breakdown the stable cobalt intermediate. My main contribution to the project was synthesis of compound 11, one of the amine pendant series. I synthesized it in 7 steps from commercially available starting material. Unfortunately, I don't have enough time to explain the synthesis in detail. Luckily, my compound was the best, and I'm going to focus on it. As before, we tested DNase activity. For 11-copper, the activity is the same as 4-copper. The amine doesn't affect. But for 11-cobalt, we see significant activity even at micromolar concentrations. Means the amine enables and enhances the catalysis. Then we tested the vulnerability. We added 100 millimolar glutamate and checked the activity. While cobalt cyclane is inactive at 0.1 millimolar, 11-cobalt is still active even at 0.005 millimolar. This is a proof of concept that the amine cap can protect the metal center. Then we checked the MIC. For 11-copper, we do not see difference between the metal-free and metal complex. Again, means the amine doesn't affect. Probably the amine doesn't function as a cap. But for 11-cobalt, we see significant improvement in the MIC, up to 8-fold. Means that 11-cobalt performs DNase activity under physiological conditions, actually overcomes the vulnerability. When we added DTT, we can see high improvement, up to 64-fold, indicating the high oxidative potency of 11-cobalt. For conclusion, before my joining, the group found out that 2-copper and 4-copper can cleave DNA at the ternary complex. They also discovered the vulnerability. The water ligand exchange under physiological conditions that today activates the complexes. Now, we saw that 11-cobalt has an amine cap that protects the metal center and enable catalysis under physiological conditions, overcomes the vulnerability. Thanks all, and thank you.